Good afternoon. Very happy to be here. Hope you all had a nice lunch. I, for one, was again very happy with the lunch. So today I would like to talk to you a little bit about my research on intestinal immunity, and I use zebrafish as the model of choice, and I will tell you why. So first of all, I'm from Wageningen University, so the people that were in the youth program already saw this slide. So Wageningen, and also was explained already by Dr. Kormelink, that showed that our campus is big and it's expanding. We have 6,500 staff members and about 10,000 students, of which they come from hundreds of different countries. And this is mainly because also our master program is in English. So it's a very international environment to be in, which I like very much. Oh. So actually, I'm located on the other side of these buildings, actually here, in the Zodiac building, in the Cell Biology and Immunology group of Animal Sciences. So where is Wageningen? Well, it's in the Netherlands. And here you see the map of the world. Well, can you see the Netherlands? Not really. So if we zoom out to the European Union, you see this little green dot here. Well, this is our country. And basically, we have 17 million people. We consider our country to be very crowded. But if you then think about Tokyo with 38 million people, I have to reconsider. Our country is not crowded at all. So if you travel from our country from north to south, it's only 300 kilometers. And from east to west, only 200. So basically, most of you would consider the Netherlands as one big city. So I have been enjoying already for quite a while now, so I've been here already 10 days. And I was very happy with the hospitality I have received here from you. So especially Mutsumi and her husband Kyohei really took me on a tour to Matsushima. One of the first days I was here, which was really great. So we ate very nice fresh fish. I loved it very much. Of course, we had a very nice youth program, which was also I enjoyed a lot because of the interaction with the students, even dressed up entirely Japanese style, discussing the posters, but also visiting your beautiful country and the culture. I think so far it has been so great. And after I leave here, I will also travel around, so I will see much, much more. So it's not over yet. It's very nice. But so I really am now a Super Saiyan because of all the nice things I've been seeing here. So now go to the scientific part. So what my passion is in research is the crosstalk between the microbes and the host. And basically, what I try to look at is this interesting surface, basically of the intestinal surface. So here you have only one cell layer between the outside world, basically, and a lot of bacteria and feed, and the human or the animal body. Of course, there's a lot of immune cells making sure that everything stays okay, but there's a very intense crosstalk going on. So in my talk, I will try to address two same sides of the coin, I would say, or other sides of the coin. The first one being that the microbes can determine the mucosal immune system. So it can actually educate the host. And the other part is that the other way around works the same. So you also have the mucosal immune system itself can influence what kind of bacteria can survive in the intestine. So first, this first part. So what has been done already more than 10 years ago now were the first studies using zebrafish as a model. And of course, using fish, we didn't quite know, because coming from a mammalian background, using the fish more as a model for human disease, we actually didn't know whether microbes and the response towards microbes was the same in fish as you would see in mice and humans. So we knew in mice that there was a lot of education of the microbes of the host immune system, but we didn't actually know so much about the zebrafish. So this is what we tried to find out, and there was this Dr. Rawls in North Carolina. He was the first actually to start using zebrafish and making them germ-free, and then colonizing them with different microbes to see what happens to the host. And what he found was that just like in mice and mammals, you see that the epithelium, so the cell layer of the intestine, start proliferating upon the microbes. So you see development of the intestinal surface, you saw turnover of these epithelial cells, and that doesn't really happen if you look at the germ-free. Next to that, he also looked at the transcriptional response. So if you colonize zebrafish that do not have microbes with microbes, and he compared what kind of genes were upregulated or downregulated, he found that almost 60 genes were shared between mice and zebrafish. So genes involved in immune system, but also metabolic processes, indicating that also in fish, the colonization by microbes that happens naturally will induce a host response. So what we see is you see that the intestines will develop, so you see an increased epithelial differentiation and turnover. You see that the specific immune genes are induced, and we now know that it makes a difference what kind of bacteria you colonize them with, what kind of genes are upregulated. And as we know from mice studies, is that it also makes a difference if you have one bacterial species, what kind of immune cells are recruited. So that actually leads to the following, is that if you have a different microbial composition, you might also have a slightly different mucosal immune system and a slightly different amounts of cells present. So this idea is actually that maybe the set point might make some individuals more susceptible towards disease, while others might be a little bit more resistant, just on the basis of what kind of intestinal milieu, if you wish, is present there upon colonization. Also, it has been shown by others using mice specifically, is that it may be age dependent, so this effect of the microbes is different in early life compared to adult life. So what they have shown in mice is that when they colonized germ-free mice at an adult age, it could not change certain immune cells that have been recruited, while they gave the microbes at a neonatal age, they could. Meaning that in the early life, there might be a window in which these microbes can change gene expression, while if you look later on, this program might not be available anymore. And they also showed that it had to do with epigenetic effects on certain chemokines. So, but having all this literature in the background, we actually started using zebrafish to see whether we could make a model for human intestinal disease. Because if this is true, if you have different colonization and you have a different susceptibility, we might be able to show that in a model system. And this is actually what I was working on at that moment. So I started my first postdoc at the Erasmus Medical Center in Rotterdam. And I worked for a clinician who was a gastroenterologist who said, we need a much more simpler model. Let's go to fish. Well, I've regretted that sentence a lot of the times. The fish are not a simple model. So, of course, there are mice models available. They, of course, have a lot of tools in mice. While in zebrafish, a lot of the things, especially the immune system, was not that known yet. So we had to figure out a lot of the things by ourselves. But inflammatory bowel disease is a chronic intestinal inflammation that can occur in humans. And a lot of the genes that are associated with the disease have something to do with the combination of the host with bacteria, so the interaction between the host and the bacteria. So a lot of people have said, okay, so maybe this dysbiosis, so the disturbed balance between the microbes and the host, might be contributing to getting either this ulcerative colitis, which is taking place at the end of the gut, or Crohn, which really can take place throughout the whole intestinal part. So, and since we know that it's not as clear, you have ulcerative colitis, but you have Crohn, but you have also many more subtypes in between, we thought maybe if we could use the fish, we might be able to distinguish different subtypes of IBD. So, but not only, so now, not only when we think of human models, also fish and other mammals can get enterocolitis, can get inflammation of the intestine. So one example is that they started replacing the fish meal in the diet of salmon with soybean, because there was a high demand for fish meal, and of course the fish grew very good on it. The only problem was that you have to feed a lot of fish meal to get, so for 1 kilogram of salmon you need 1.4 kilograms of fish meal, so that's not really sustainable. A lot of the fish meal was used by the aquaculture industry, so the prices go up a lot. So the industry is looking for other sources, and they came to soybean meal. But what they realized is when, so the fish that stopped growing and they were not so happy, and they realized that salmon can actually develop enteritis due to the soybean. So if you have high inclusion of soybean in the diet, you get damage to these are the intestinal folds, so the villi that enlarge the surface of the intestine. You see that they get damage here. So after 5 weeks already of feeding soybean meal, these animals have problems taking up nutrients, mainly in the last part of the gut. And many more fish species now have been shown that to be susceptible to this. So there's a lot of studies being done, and the different anti-nutritional factors that might cause this problem. But also here we are in the future now, or just awarded a grant to be able to look at this and using the zebrafish as a model. So this is indeed what now my aim is. So we can develop a novel model to study intestinal health and disease. We can use it maybe as a model for human disease, but I think also we need to know much more about the fundamental processes before we can even begin to think about asking the right questions in human disease. And we can use it as a model for aquaculture fish species. And indeed, we would really like to know the fundamental processes. So when do what immune cells recruit to the gut? When do they react? To what do they react? So a lot of unknowns. So that makes my job really nice, because I have so many questions I can answer that I will be in business for the next 100 years. So this is basically what we started out with, and this seems a little bit harsh, and it is. First we thought, okay, we need to have some sort of model for intestinal inflammation using zebrafish. And since zebrafish have first in early life have innate immunity and later in life develop adaptive immunity, we started actually by using adults. Because in the adults you have both innate and adaptive immune system. And for a lot of the chemically induced enteritis models in mice, they use haptens, and they're considered to work via T cells. So we wanted to know whether if we would inject these fish with a chemical named oxazolone, which is a hapten, if they would get a sort of an intestinal inflammation, if you could see that. So what we did is we transferred our fish to a standalone tank 24 hours before the experiment, and then at T0 we injected intrarectally. We injected some of the oxazolone or its control, which is a high amount of ethanol. So of course that will also give damage. The oxazolone needs to be dissolved in ethanol, so it's a very harsh treatment. And then after a couple of hours and 1 week and 2 weeks after, we collected the tissues and the cells to see what was happening. So this is basically what we did. We devised a score to look at the histological damage that we see. So here we see healthy intestines of the fish. So you see nice, maybe you cannot see in the back, but there are some purple or some pink cells right at the bottom here. These are the eosinophils. This is an Alcium blue pus staining, which stains the eosinophils bright pink. And then you have these goblet cells secreting the mucus. So they make that mucus layer on top that are purple and blue. So you see the nice folds, a very tight lamina propria here in the villi, and the epithelial cells nicely aligned. And the things that we could observe was the bowel wall thickening, so really thickening here. And well, you see also the villi here do not look very normal. You could have loss totally of these villi after treatment. You could have goblet cell alterations, so either a lot of goblet cells, we also observed that, but we also observed that they were just gone because of the damage. And we had a score for the infiltrating eosinophils. You can see that here they are basically at the bottom of the villi, while if you look at some of the colitis scores, you saw that they really move upwards into the villi, responding to the chemical. So then we quantified this, of course, and then we see this. So the gray bar here is the baseline, and this is the score of all these different parameters that we set a score to. And then you see, of course, when you inject ethanol, which is a very high concentration, 50%, you see that indeed you see damage. But that is resolved at 1 week. You don't see any damage anymore after 1 week. While if you look at the oxazolone treatment, still after 1 week, you could still see a higher score compared to baseline levels. At 2 weeks, this is gone. Of course, this is not a model for chronic inflammation. It's more an acute damage model. That also holds true for the mice models, because also there, within 10 days, you don't see this effect. So what we mainly study here is effect of the early damage. So, but of course, we were wondering now, does this enterocolitis development depend on the microbes that are there? So what we did is we repeated the same experiment, only now we added 2 more groups. So what we did, we gave colistin, so an antibiotic, in the water, or vancomycin. And it was just not to specifically knock out some species, but really to skew the microbiota one side or the other. And then we repeated the entire experiment to see what happened. So first of all, did we change the microbes? Well, yes, we did. And this was early days. So this is cloning and sequencing. So we had a lot of mini prep Fridays, as we call them, by getting all these sequences out of the E. coli and sequenced. Of course, it also means that we sequence about 100 clones. So it's not as thorough as a really 16S sequencing we can do now. But what we basically see is that first thing we see is that the diversity goes down of the species that are there, which you see a lot when you give. So this is after the treatment with oxazolone. But you also see in the case of the colistin-treated animals that you see more Aeromonas species growing out. Well, if you give vancomycin, you see that the Cetobacterium somerae, which is a fish commensal, was growing out, which was interesting. So indeed, we knew at least we have shifted the microbial composition. But now, of course, we wanted to see whether we had any effect on the enterocolitis. So I only show you here the data of the 5 hours. So the acute damage, which was most striking effect. And first, well, let's just, now I spill the beans. So what we see here is that if you only give the baseline, so just baseline or just the antimicrobial substances, you do not change it. Well, this is of course not expected. It's really short treatment. It's only 24 hours, 48 hours before you start the experiment. So it's not expected that you have the time to get outgrowth of pathogenic bacteria or anything. But if you look at ethanol, you still see that there's a little induction. Oxazolone gives the higher induction. But then if you see the fish that are pre-treated with colistin, they show about the same level. So it's not significantly different from the oxazolone model, while the fish that got the vancomycin actually did not show any damage. So here we could see, and even after 24 hours, although it was a bit higher, so it went up to 7 at 24 hours of vanco, it never got as high as the 20 score that we see here. Meaning that apparently the microbial composition has an effect on the severity of the enterocolitis. So to summarize this, so we see if we give vancomycin exposure that we change the intestinal microbial composition. We see a domination of the fusobacteria, and it protects the fish from these early time point damage in enterocolitis. Also, due to time, I did not show this, but we see a specific reduction in the recruitment of neutrophils to these fish in the vancomycin treated, while you don't see that in the colistin. Colistin did reduce the eosinophils, but vancomycin reduced both neutrophils and eosinophils. So meaning it has an effect also on the severity. So thinking about this, then now we know that the microbial composition has an effect on disease susceptibility. We can also think about aquaculture fish. So if we know now that there's a lot of antibiotics pollution in our environments, you might also see that some fish will maybe are more susceptible, or even more resistant, but also more susceptible towards inflammation. So a very important point to know what kind of microbes are in there. So the second part is basically the other way around. So what does the mucosal immune system do with the microbes? And here, of course, there are a lot of factors determining what kind of microbes will eventually colonize. So of course you have the environmental bacteria, because of course the bacteria that come into your gut are in your environment are the ones that are able to colonize or not. Another important host factors is the pH, so the niche, the kind of nutrients that are present, but also the kind of diet we eat, the feed the fish get. But a very important additional factor is also the host immune system. So the antibodies produced, the cells present can also attack certain species while it leaves others alone. So an interesting experiment was done again by Dr. Rawls already in 2006, in which he was interested in what kind of host, does the host affect microbial composition? And he did this very neat experiment, I still like it very much. So what he did is he actually transplanted the microbes from the mice to the fish, so to germ-free fish, and the other way around. So he also took fish microbiota and then put that in a mouse. And then saw over a couple of weeks, checked what was able to grow out in these mice and in the fish. So, and I'll take you through this data, because here on top you see the conventionally raised zebrafish. So this is a zebrafish microbiota in phylum level. So you'd see it's mainly proteobacteria, it's mainly the red. And if you look at the mice, they have mainly firmicutes. But if you now look at the zebrafish that get the mice microbiota, you will see that these red will grow out. Also the ones that are able to grow to dominance here. And the vice versa, what happens is if you get the mice with the zebrafish bacteria, you see the zebrafish bacteria contains almost no firmicutes, but the ones that are there are able to grow out. So this means that the host actually shapes the microbial composition, and it will shape according to what the host accommodates. And of course, these are more factors than just the host immune system. It's also the nutrients, it's also the niche. But this actually determines quite a lot, which is very interesting, I think. So the host factor immune system, the immune system comes into play. So to reach homeostasis, the host must be able to mount an appropriate response towards pathogens, but also should leave the commensal bacteria alone, because these commensal bacteria are very important to digest our feed and to provide certain nutrients for the host, and also protect against some pathogenic species. So it's very much a very dynamic ecosystem. So what is known from mice, from the immune system, what happens in control of the microbes? Well, we know that in mice you see that a lot of the deficiencies in the innate bacterial receptors, so the NOT, so the internal toll-like receptors, so the MyD88 deficient, they all show differences in their microbiota, meaning that these processes play also a role in shaping. The other thing is also true for the adaptive immune system. So the adaptive immunity, such as IgA, immunoglobulin A, is also affected, so affects the bacterial composition. So if you have no IgA, you will have overgrowth of certain bacterial species in mice. Then you have all sorts of other models, so the CD1D deficient mice. Let's go back. So the CD1D deficient mice that do not have NKT cells, they will show a different microbiota. The RAC1 deficient mice harbor a different microbiota, and these mice do not have B and T cells. And that holds true for either B or T cells. So innate and adaptive immunity both have an influence on microbial composition. So the nice thing of the zebrafish model is that we can look at this sequential development of innate and adaptive immunity. Because in the first week, you will have only innate processes. Of course, these fish grow from eggs, and these eggs are directly exposed to the environment. And then they only have the innate system, which we have already heard from other speakers, that it's very powerful. Plants only have innate immunity, so they can handle a lot. The insects also, same case. So the innate immune system here in the first week can also handle a lot. Then slowly over time, adaptive immunity develops. So the B and T cells will develop in the zebrafish. And then at adult age, you will have both innate and adaptive immunity fully developed. So that makes it an ideal model to study the difference in microbiota at different time points. So my supervisor at that time did not really want me to go into the effect of adaptive immunity, but I finally convinced him that we should really look into it. And I asked him, okay, just let me sequence the REC knockout, so the fish without B and T cells, compared to the wild types. And just take adults, a few fish, and we'll see if the microbiota is different. If the microbiota is not different, I will stop this project. But if it's different, I would really like to carry on. So he finally said, okay, go ahead. So I was thrilled that I could do it. And I started to go ahead and sequenced. And this is then what we observed. So here is a PCA plot. So here you see the microbiota. So every dot here is a fish. And these ones, the homozygotes, these are the REC knockouts, so not having B and T cells. While these are actually the wild types, the green ones, and the heterozygotes. So you see these microbiotas really cluster together, while the REC knockouts really appear to be different. So I was very happy with this result, especially since I have only sequenced like 4 fish per group. So the effects were pretty big. So when we looked at species level, you saw the microbiota was not as diverse. So you see that there are different species present, but mainly there was a dominance of this green bacteria. And this was actually the Fusobacterium, so actually also the Cetobacterium somerae that we already encountered when we gave like the vancomycin that grew out. But what was clear is that we have 2 groups, and the REC knockouts actually show more of these brown species, which appear to be Vibrio. And of course we know that Vibrio contains a lot of the pathobionts or pathogenic species. So apparently they have a different microbial composition. So then I would like to look at these different time points and see whether I could look by qPCR for these different groups, whether we can follow the colonization over time in these fish to really address these different periods. So only innate, adaptive immune system development, and both innate and adaptive immunity. So we designed qPCR primers and looked at Fibrio and Fusobacterialis, so the 2 dominant groups, to see what was the difference. And then I analyzed it at 1 week, so only innate, at 5 weeks, so in the middle of this development of the adaptive immunity, and at 14 weeks of age. So and this is what we then observed. So specifically if you look at the Fibrio, so you have to realize that at 1 week post-fertilization, these fish, the wild types and the knockouts, both have only innate immunity. So they are the same. And luckily for us, indeed, we see that the microbes are also the same. So you see that the Fusobacterialis and the Vibrio are about the same in these larvae, in levels. But then the difference occurs at 5 weeks. So the wild types, apparently, the Vibrio goes down, while the REC knockouts, they stay high in abundance. So somewhere during, we think, this adaptive immune system development, you see that these fish are able to reduce these Vibrio inside the gut. So of course now we wondered what kind of cells are doing this, because the REC knockouts have no T cells but also no B cells. And at the moment we only had the T cell reporter and not the B cell reporter. Nowadays we have an IgM B cell reporter. So more studies are underway, but what we have decided to do is to isolate the T cells from our LCK T cell reporter, which you can see here. So here you see the thymus containing these green T cells. So we were able, by a flow cytometer, to really sort the lymphocyte population into GFP positive cells, so the T cells, and the GFP negative cells, which will be the B cells and probably also some NK-like cells. Then what we did is we adoptively transferred it, a few cells, only 15,000 cells, and put that in the REC knockout fish. And then 1 week after transfer, we checked whether we could find these T cells. We had no idea if they would survive or would they just be killed by the other fish, or we had no idea, and also to see whether the microbial composition would then change. So would we then see a shift in this Vibrio bacteria? So this is to show that indeed we see, so we did several experiments and we see that in most of the fish we could find a GFP signal back in the intestine after 1 week after giving these cells. So what means that indeed the T cells were able to at least survive for a week, and we could find them in the intestine, which was very surprising at first. So we had a sort of a clue, well, maybe then if they were there, maybe they did something with the microbes or not. And yes, indeed they did. So if you do, these are REC knockout fish, either given PBS injected the week before, or given this non-T cells, so the GFP negative fraction, which would contain the B cells and the NK-like cells, or the T cells. And then you clearly see that the T cells were able to, after 1 week already, you see less Vibrio getting around to the amount that you see in the wild types. Meaning that these T cells are, in one way or the other, able to reduce these Vibrio. So the other thing, and that's usually when you start doing the research, you find sometimes some weird data, and you think, oh, this cannot be true. I don't get this. So you leave it for a while, and then after a few times you repeat the experiment and find again this strange data. So it has something to do with your experiment. So I had the same experience when I checked the different cytokines over time in these REC knockout fish. So I checked different cytokines to look at the intestinal milieu to see how these fish were doing. And we looked at different cytokines. So we looked at IL-1 beta, we looked at 2 types of the IL-8, IL-17, interferon gamma, IL-10, and most of the cytokines you see basically at 14 weeks, so at the adult age, you see some difference. So you see more IL-1 beta in the REC knockouts. You see less IL-17, less interferon gamma. Well, they don't have adaptive immunity, so that makes sense. Less IL-10 also. You also see that these older REC knockout fish are not that healthy, so that is very reflected in that you see more IL-1 beta, more IL-8 like 2, because they have 2 forms, and also less IL-10. But this very strange data point keep popping up. So there's this IL-8 like 1, which was only high in the wild types at 5 weeks post-fertilization, and we just couldn't find out why this was. It was really strange. So we decided to look if this correlated to the existence. If you look in wild type, do you see then that it goes the same with the LCK GFP cells? So if you have T cells coming into the gut, does that correlate with this time point? Well, not that clear. So when we looked at the T cells presence in the gut of these fish, you see that from 3 weeks you can pick up about 5% of these cells in the intestine. So meaning that the cells, at least T cells, are there at the moment that this peak was there. So then we decided to look in our adoptive transfer experiment to see whether this IL-8 like 1 is also changed. And indeed, that was the case. So not this like 2 form, that was about the same when you inject. You get some increase in both the non and the T lymphos. It was really low expression levels as well. But this one, you see that if you give T lymphocytes, you saw in the intestine, you see a lot of IL-8 like 1. So maybe the T cells themselves produce this IL-8. It has been shown in mice and in human cells that T cells can sometimes produce IL-8, which is a potent neutrophil recruiter. So here you see like the correlation between the LCK and the IL-8. Well, that's quite convincing. I would say that maybe it's just the T cells themselves producing it. So we also looked at sorted T cells and compared them to sorted neutrophils. And we indeed see that if you sort the cells and then looked what kind of cytokines they produce, you see a high level of, well, a relatively high level of CXCL8 like 1 in these T cells. And not so much in the neutrophils. The interesting thing is that this was not so high as what we saw in the entire intestine. So still we thought, might other cells also produce IL-8? Because it's known that epithelial cells themselves can also express it. So then we went to in situ hybridization. And what we then observed was that the epithelial cells, so this is RNA expression in the intestine, you see that if you have the wild types at 5 weeks, you see that the epithelial cells and some scattered cells throughout the intestine produced or expressed this IL-8 like 1. While the REC knockouts, you see some scattered cells, probably the granulocytes, that produce this IL-8 like 1. But then, if we give back the T cells to these REC knockouts, this is what we saw. So basically, these transfer of the T cells made this IL-8 production in the epithelial cells, or the expression, go up. Which is, I think, a strange way, because we always think innate, so the epithelial response of IL-8 is something that is not controlled by adaptive immunity. We never thought that way. So to summarize this a little bit, so the T cells can modify the microbial composition by reducing the abundance of Vibrio species. And this IL-8 is induced in epithelial cells after adoptive transfer. And this might be a conserved mechanism, we have no clue. But also we don't know why this induction of the IL-8 can specifically reduce Vibrio. Why would that be specific? It's not real specific signal. So we will be looking into this in the future. We have a lot of questions, more questions than we have answers, I think. But in the last couple of minutes, I would also like to address my ongoing research. So the things we are also doing next to diving into the more fundamental mucosal immunology. And this is to look at this nice larvae that we have. And this is always a nice way of looking at the zebrafish. So if they're very small, so up until 3 weeks, you can see through them. So you can really anesthetize them, mount them in a solid gel, and then you can do live imaging. So you can really see the cells moving in the entire animal. So here we have a transgenic fish that has green neutrophils and red macrophages. And if you have recruitment or inflammation in the gut, you will see that they will come closer together. So they will cluster. So here you see this yellow light here is the red and the green overlapping. So this might be a nice way to really study how the immune system responds to, for instance, feed ingredients. The other thing is what we can do with these fish is to look at live epithelial barrier function. So it's like a little bit of the Smurf phenotype of the Drosophila. So here you actually let them swim in a solution containing dextran fits. So it's a sugar molecule with a fits label, so a green label, and it will be ingested. But once there is leakage of the barrier, you will see that this green substance will go out of the fish, or will go inside the fish. And you can really measure this. So then you can go for live barrier function. So the other thing, of course, that we can manipulate this fish quite easily by using the CRISPR-Cas genome editing system. So if you would inject CRISPR-Cas in the one cell stage, you could modify host genes involved in mucosal immunity and then look at the effect in these fish. And of course, my hobby also is here the microbial composition. So we are in the process of trying to create germ-free fish and then colonize them with different species and have a sort of a test tube to really address different bacteria. What do different bacteria do with the different immune cells? So of course this work was not performed just by me alone. It's of course a lot of work, and of course for the students now it's a very nice story if you look in hindsight, but it has also been a lot of hard work and a lot of experiments failing, right? So if we give these talks, it looks like everything was happy and good all the time. But of course this is not the case. So this is hard work of a lot of students, a lot of colleagues of mine. So in the cell biology and immunology group where I am now, I'm greatly indebted to Maria and Geert, and Huub Savagul is the chair. I work together with microbiology and host microbe interactomics with Leiden University, who are very skilled in zebrafish work. I've worked at the University Medical Center, where all these colleagues had a big role. The University of Sheffield, so Stephen Renshaw, who made the neutrophil reporter fish. The Hubrecht Institute, which is very well known, using zebrafish as a model for developmental processes. And of course, I started out first at the Erasmus Medical Center in Rotterdam. So with that, and also thanking our funding, I would like to end, and I would take any questions that you might have.